Hi there. In this video we'll discuss central metabolism, which as we saw is the two processes of glycolysis and the citric acid cycle. Glycolysis converts glucose, which is 6 carbons, into 2 molecules of pyruvate, which is 3 carbons each. This occurs in the cytosol. It's anaerobic and it produces a small amount of ATP. The pyruvate will then be transported into the inner mitochondrial matrix. It'll get converted to acetyl-CoA. Acetyl-CoA will enter the citric acid cycle, which is also referred to as the TCA cycle, the tricarboxylic acid cycle, and the Krebs cycle. All these names mean the same thing. And once, as part of the citric acid cycle, you will burn 1 carbon of acetyl-CoA at a time. There's only 2, so it's going to do it twice. Generate high-energy intermediate molecules NADH and FADH2. And these molecules, which we'll see in the next set of lectures, in the next class session, these produce a lot of ATP in an oxygen-dependent manner in a process called oxidative phosphorylation. This is a reminder of the overall pathway which we'll cover today. Glycolysis is the conversion of glucose to pyruvate, and the citric acid cycle is pyruvate being converted to acetyl-CoA and getting integrated to this cycle and producing carbon dioxide and NADH and FADH2, which goes on to make a lot more ATP. Now here's a bit more detail on this process of glycolysis in which glucose gets converted to pyruvate. I need to mention that I've made the decision not to require you to memorize the structures of the carbohydrate, even though I think most similar courses which cover less material would require. It's also essential if you plan on taking the MCATs because they will test for the independent structures. But for this course, this slide will just look at the process at a higher level. Note that the first thing we do is we're going to invest 2 molecules of ATP. So if the goal is to produce ATP, we're already 2 in the hole right off the bat. These 2 ATP molecules end up donating phosphoryl groups to the sugar, and we get fructose 1,6-bisphosphate, which is then split into 2 3-carbon sugars, initially different, but then they are both converted into 2 molecules of glyceraldehyde 3-phosphate. And now from the aldehyde moiety, we're able to make NADH, which will provide dividends down the road, and we'll be able to make 2 molecules of ATP from each of the 3. And so a total of 4 ATP molecules are made, minus the 2 we've invested, means we have a net of 2 ATP during glycolysis. This is what you get from anaerobic metabolism. You'll see that oxygen has not entered into the picture yet here. The process of making ATP without oxygen is referred to as substrate level phosphorylation, as opposed to oxidative phosphorylation, which we'll see occurs in the mitochondria. In oxidative phosphorylation, each of these 2 NADH molecules will be worth about 3 ATP each. So we've made basically 10 molecules of ATP here, although 6 will be coming later. And then finally, pyruvate is the molecule that will be shuttled into the mitochondria for the citric acid cycle. I'd like to look at a detail of step 3 in glycolysis. As you know, we invested 2 molecules of ATP. The first one produces fructose 6-phosphate. At this point, the cell can still use this molecule for things other than glycolysis, or it can be converted back into unphosphorylated fructose without significant loss of energy. But now when we invest this second molecule of ATP and use the enzyme phosphofructokinase, now we're committed to pursuing glycolysis with this molecule. This is referred to as the committed step of glycolysis. And that's because we've invested so much energy now into the production of that molecule that it's too wasteful to do anything else with it. So here's some detail of steps 6 and 7, which are some of the energy producing steps. See, here we're making NADH, and here we're making the first of 2 ATP molecules. So these are energy capturing steps. Notice that we have to, the first thing that happens is we utilize a molecule of NAD+, which we're combining with glycerol 3-phosphate to form this high energy intermediate. And normally from that NAD+, we're going to get the NADH, which in the presence of oxygen is a good thing. If oxygen is not present, like in anaerobic situations or when you're exercising and you can't get enough oxygen to your muscles, the important thing is having plenty of NAD+ present just so you can proceed, not so that you can get energy out of it. Now notice that we've taken an aldehyde, which is a highly reduced form of the carbon, and converted it into an intermediate of a hydroxyl group, and here it is a thioester, which is an oxidized version of an aldehyde, but this is still a high energy bond, and that can later be used to form a high energy phospho intermediate, and this phospho group can be transferred from ADP to ADP, forming ATP in the process which I mentioned is called substrate level phosphorylation. And on this slide we can see the results of those two steps summarized. We have an aldehyde converted to a carboxylic acid, which is an oxidized form, more oxidized than aldehydes. We've gotten high energy electrons in the form of NADH, and we've captured some of the bond energy as a molecule of ATP. And on this slide, although making NADH and ATP themselves are energetically costly, we show that steps 6 and 7 overall is energetically favorable, the delta G of -3 kilocalories per mole. The oxidation step drives the synthesis of NADH and the formation of this high energy bond, which can then be hydrolyzed to produce ATP. So this has gone on in the cytosol. The sugar has been converted down to pyruvate in the process of glycolysis, and what we'll see in the next slide is that fat's taken up from the bloodstream, fatty acids imported into the cell. They'll go straight through the cytosol into the mitochondria, and they'll get converted to acetyl-CoA as well, allowing the citric acid cycle to occur. Once fats and sugars are converted to acetyl-CoA, the metabolism is the same for the both. How could it be any different? The conversion of pyruvate to acetyl-CoA is a pretty interesting process. Again, pyruvate is the 3-carbon molecule. That's the end point of glycolysis. Acetyl-CoA is a 2-carbon molecule. So we're going to lose a molecule of carbon dioxide. This represents the first loss of carbon in the process. And 3 different enzymes are involved with the production of NADH, the formation of another high energy intermediate, eventually producing this molecule called acetyl-CoA. What's interesting about it is the structures of the enzyme complex pyruvate dehydrogenase, which has 3 different subunits, subunit A, B, and C, which are doing 3 separate reactions. Now we normally can think of these reactions occurring in solution, where one enzyme would work on a substrate, it would release the substrate into the solution, it would diffuse around, it would hit another enzyme, and it would go ahead and do its biochemistry, but that takes a long time to encounter the substrate and the enzyme. And what actually occurs is more like a bucket brigade, where one enzyme, the blue one, will hand off the molecules directly to the second enzyme and will hand it off directly to the third enzyme. The substrates are never free in solution. They are not given a chance to diffuse away. So this is a very speedy way of performing 3 successive reactions in a geographically constrained area. Now here's a molecule of CoA. The acetyl group would be added right here on the sulfhydryl group. You do not need to know this structure. I just wanted you to see it. You'll recognize the basic structure here of ATP or ADP, and then a linker section. It's almost never drawn out. It's always just represented as like this, acetyl-CoA, but I just wanted you to see it once. Now to conclude this portion of the video, I just want to get ready for the citric acid cycle. So I want to talk about the breakdown of fatty acids in the cell. Remember, the fats are absorbed from the bloodstream. They're converted to fatty acids. They are transported through the cytosol right into the mitochondrial matrix, and then this cycle will occur. This is called the beta oxidation cycle of fats. It is not to be confused with the citric acid cycle. The reduction of the two carbons occurs in 4 steps. And we're starting with an energy-rich saturated hydrocarbon, which is a single bond. The first thing that's going to happen is we're going to eliminate 2 hydrogens, forming a double bond, which will then get oxidized by the addition of a hydroxyl group, which will get converted to a carbonyl. Then we'll cleave this bond, releasing acetyl-CoA. Now these 2 carbons are just like the acetyl-CoA produced by glycolysis, and we'll reattach another molecule of CoA to the now shortened fatty acid. And we're going to repeat this cycle as many times as it takes to cleave the hydrocarbons down by 2 at a time until we have cleaved them all, we've captured them all as molecules of acetyl-CoA. In the process of making acetyl-CoA, which will be very useful for oxidative phosphorylation, we're also making molecules of NADH and FADH2, which will also be able to be cashed in for molecules of ATP down the road. And here's where we'll start the next lecture with acetyl-CoA, whether it's produced by glycolysis or by beta oxidation of fatty acids, entering into the citric acid cycle. Thanks.